So I'm going to skim over some of the historical background, but I think if we dial the clock back to the mid-1800s is when animal experimentation began, and Leslie articulated the ethical opposition to that, which sprang up pretty much at the same time that they began vivisecting or live dissecting animals in the town square, no pain relief. This is the earliest beginnings of animal experiments, and there's always been an ethical opposition and an anti-vivisection movement for as long as this industry, this activity has existed. If we dial forward now to the mid-1950s, another school of thought began to appear, which is the scientific case, to say, you know, mice are not little humans with tails. We're different species, and an extremely hard-line scientific argument began to be articulated saying, you know, the use of the word scientific fraud has been used. And then a third school of thought also emerged in 1959, a response from the scientific community itself, and the principle of the 3 Rs, to reduce, to refine, and ultimately replace the use of animals in science, was also articulated. And it doesn't really matter which of those resonates with you, because I think if we look at this in the Canadian context and look at the straight numbers, the statistics on animal use from 1979 until a couple of years ago, you don't need to be a statistician to see the trend line is going up, not down. And although these are summary numbers, they don't capture some of the victories that Liz and Leslie have pointed out. On the whole, we're not winning, and the animals are not winning. And this is despite the 60th anniversary of the 3R principle from the scientific community. I think if you go to any institution, U of T, U of Guelph, and ask scientists, do you support alternatives? Do you support the 3Rs? Everybody does. And yet, why is the line going up and not down? This is also, you know, CCAC, which Leslie introduced, has been around since 1968, decades, 50th anniversary of this institution, which is supposed to be enforcing the 3R principle through Canada. Again, the proof is in the pudding. So I hate to start on a bit of a downer, but I think we need to be realistic about where we are in the state of play. I entered the world in 1975 and the animal protection space in the early 1990s. And again, you can see there have been dips in the numbers, but overall, they're going up. And it's really forced me, as Rob suggested, to take a step back and really scrutinize, are we just doing the same thing again and again and expecting a different result? And are we doing a disservice to the animals in so doing? I don't know whether Einstein actually said this. This is a Google image, but whether or not that's the case, some wise person did point this out, and I think we do need to take that to heart. I don't know if you can see a buzzword that's going around the internet these days a lot is it's time to think outside the cage. Researchers, stop equating animals to humans, but I think we in the advocacy community really need to take a good hard look at, again, as Rob said, are our activities, are our strategies actually working? And if they're not, we need to course correct. Another concept that gets thrown around a lot is evolution versus revolution. So if we look at the 3Rs as an evolutionary concept, we tweak individual experimental proposals in the hope that we'll get to replacement one day. So replacing, you know, an animal test with an alternative in the idea that a one-to-one substitution is necessarily going to work, I'm afraid the reality is much more complex than that. And I think if we look backwards at another animal practice from the 1800s, which is one of the founding issues of the modern humane movement, it was anti-vivisection and carriage horse welfare. Those are the two issues that founded most SPCAs in history. And there's been a century of history of advocacy that these animals are horribly treated. We need to modify how we behave towards them, how long they can be made to work, the welfare standards that are provided to these animals. But the number one thing that actually addressed this issue, mostly once and for all, is the invention of the automobile. Coming up with a better alternative, a revolutionary way to address a human need. And if we view animal research, animal testing through the same lens, as Leslie said, it's the wrong thing, but for an acceptable reason. Curing disease is not selfish. There's nothing wrong with that. But can we have a better tool that will be more effective than pretending that rats and mice are miniature people? By the same token, if we wanted to go to Vancouver today, we could drive 45 hours, I suppose, in the automobile, or use the latest innovations. And say what you will about Air Canada service and food quality, we're going to take the plane because we like shiny new things. Every other aspect of our society is embracing hungrily the latest technologies, the latest innovation. Why are we still dealing with animal model principles from the 1800s? What's wrong with this picture that this is the one area in our lives that is so firmly entrenched in the past? I hope you're going to be able to see these next couple of slides, but I think it's important to really understand the problem and understand the language that we use. We tend to use animal research and animal testing interchangeably, but they're different animals. So if we begin with testing, one of the questions to Leslie was, what's animal tested? Can you give me a list? It could be pharmaceuticals, it could be vaccines, industrial chemicals, food additives, pesticides. They're all animal tested. Why? It's the law is the short answer in most cases. Before marketing a product under Canadian, European, American, Chinese, everywhere, legislation, companies are required by government authorities to submit test data to substantiate, is it safe? Is it effective? Those are reasonable questions because we all, when we go down the supermarket aisles and we pick something up, we assume it's going to be safe. We don't ask ourselves, how do we know that? What goes into that evidence of safety? Depending on the kind of product and the particular piece of legislation, there could be a long list of testing requirements. It could be, you may be familiar with the LD50, the lethal dose 50% test. They have oral skin exposure, forced inhalation variants of that test. Eye and skin irritation, skin allergy, does it cause cancer, birth defects? There's a long, often tick the box list. And this is just in Canada. Then we have to consider that we're living in an age of global trade. So it's not just Canada. If I'm a company and I want to sell any new chemical, I have to consider what are all of the laws and regulations in every country where I want to sell this thing. And if we have an alternative method, say a skin model that uses human 3D tissue instead of blinding a rabbit, if that is accepted in all but one major market, and I want to sell in that market, that country, guess what? I'm still doing the rabbit test because I have to follow the law or choose not to sell in that country. And multinationals don't become multinationals by going down on the other side of that issue. So the reality is these companies are following legal requirements which are extremely complicated and tick at least a dozen major national sets of regulations. So that's why everything is animal tested and why it takes bloody forever to change it. The other scenario is research, and that could be fundamental research. What does this enzyme do in biology? Or I would like to understand cancer, HIV, looking at a particular medical application. And here's a very different story. There is no law anywhere that says in order to investigate these things, you must use animals. What happens is there's a group of organizations. It could be a government funding agency. It could be a charity like the Heart and Stroke Foundation. It could be the Gates Foundation. Someone with money decides, I'm going to allocate this money to a particular purpose, and they determine the eligibility. So they could say, I've got $100 million, I'd like to look at lung cancer, and they publish that to the universe of scientists, and they will decide, if they don't specify, I could be looking at improving animal models, or I could be looking at human tissue on a chip And if I'm a funding agency, I could put brackets around my funding and say, no, I want $100 million for a human-based approach, or I want $100 million, we've mapped the human genome, knock some genes out of mice, see what happens. So these scientists, U of T, wherever, they're hungry. They survive based on grant funding. So they're going to be reading these grant applications and preparing proposals. And depending on, again, going back to the beginning, what are the parameters, what are the criteria that the funding body set is going to dictate, fork in the road. If I am deciding I want to do an animal type of study, I'm going to do a proposal. It's going to go to my local institutional animal care committee to judge the ethics of it. If it follows a peer review and I'm successful in getting funding, rubber stamp, dead animals. Or alternatively, if you go down the human-based track, everything downstream from that doesn't happen, and you're able to develop a human biology-based approach to addressing a human health concern. So that's just kind of a wordy way of saying a lot of what Leslie was talking about is at the right-hand side of that flow diagram. And historically, whether we're talking about the CCAC, confidentiality, access to information, all of that is happening very late in the process. I would argue too late to really make a meaningful difference on a large scale. If we can move the money and set different parameters, and think about it, if you're donating to a charity, if every person who made a donation, I won't name charities, but made it clear that they will not accept their charitable donations being used for any of that downstream animal use, guess what's going to happen? So we can make a tremendous impact at a very critical stage in the process. Looking at, back to the toxicology, the animal testing space, how do we achieve replacement in practice? And I'm glad one of the questions came up talking about the one-year dog test, which is something that HSI has been working on as well. The scientific evidence that this test, where beagle dogs, 36 per test, are broken into 4 groups, and they are either force-fed or their food is laced with the pesticide chemical for 1 year. And then at the end of the test, they're killed, dissected. It's basically a fishing expedition to see what this pesticide did to their insides. This test was declared scientifically meaningless by regulatory scientists who would know back in the late 1990s. In 2007, the US removed it as a requirement, and in over a decade later, we got the European Union, India, Canada. Last year, Japan and South Korea finally took it off the books. Brazil is the last holdout. So we actually have a situation with a company we're talking to now, which has been at the forefront of helping us get this test taken off the books. And they feel they're not able to do business in Brazil because the government is still requiring legally this one test. So it's that one holdout country is a barrier. And it's not just a barrier to one company. So how do we deal with this? I mean, this has been a 20-year process to remove one scientifically needless animal test. And there are dozens of animal tests across different regulatory frameworks, across 200 countries in the world. We need a much more efficient way of getting through this. The solution that HSI is working on is what we call mandatory alternatives laws, and it's predicated on a European model from 1986, which is it's taking the 3 R's and making it law to say, if an alternative approach is reasonably available, thou shalt not conduct an animal experiment. So you establish across the board, it doesn't matter whether it's a drug, a pesticide, cosmetic, chemical, if there is an alternative approach, the regulators must accept the alternative. And we've been successful in 2014 in having that measure taken up in Brazil, together with a 5-year mandatory phase-out for the animal test that is replaceable. A couple of years ago in the United States, we were able to amend the federal chemical law, and this deals with 100,000 chemicals per year, to require very strong animal protective language to be adopted by the US EPA and by the industry. And in South Korea, back in 2017, we were able to do the same thing with their legislation. So it's a slow process, but as we're covering these major countries whose decisions really make an impact on millions, if not tens of millions of animals every year, we're able to go in a fundamental way and hopefully dramatically accelerate the process of using alternatives. Back to Canada, cosmetics, another question that came up. This has been a long thought campaign going back to the 1970s, long before my time. But after the European Union became the largest cruelty-free beauty market, meaning banning animal testing for cosmetics, as well as the sale of cosmetics that have been subject to any new animal testing after 2013, we have seen the floodgates open, and it's by having the luxury of I have a team around the world, so I have people in Korea, Japan, China, and elsewhere. So we've been able to get 37 national bans to date, and by we, I include other organizations who also work in this space. There are 8 state bans, and we have 10 similar laws that are working their way through the legal process as we speak, Canada being one of them. So where are we with the Cruelty-Free Cosmetics Act? It cleared our federal Senate in June. It is pending reintroduction for first reading before the House of Commons. Hopefully, cross your fingers, the next week or two, we will see that. HSI and Animal Alliance have been working in lockstep on this since the beginning, and we're working to really try and negotiate with the major beauty companies and the industry position to get agreement on what the language they can live with, so that we will hopefully see this move very quickly through the legislative process. Because as you know, we have an election this year, so at the end of June, if this hasn't been through the full 3 readings and up to royal assent, we get knocked back. So I can't overstate the urgency here. I'm optimistic we're going to get there, but we really are counting on each and every one of our supporters to get on the phone, get on email to your member of parliament, and really rock the boat. So I'm going to pivot quickly to a little more science, because as Leslie pointed out, however you feel about animal protection, animal rights, the ethics, there's also the scientific reality that around 95% of new drugs that appear safe and effective in animal tests wind up being unsafe or ineffective when they're given to humans. So this, you can see the bars are the amount of money that the number of new drugs approved. The cost is the straight line. Fewer new drugs approved, higher costs. The model isn't working, and the industry itself knows it. And by the numbers, we're talking about $2.5 billion for every new drug candidate. That takes around 13 and a half years on average to come to market, and thousands of animals. And these are drug companies who gave this quote. Most of this failure is due to the limited predictive value of preclinical, aka animal models. And as much as we focus on the animal piece of it, also being cognizant, going back to Rob's idea of other communities we need to reach out to, we're all family members who are suffering from diseases. We are all not benefiting from this situation. And I think we really need to break out of your child or your dog kind of polemic that certainly some in the opposition would like us to continue going around that hamster wheel. But the reality is we're all losing, and we need to break out of that. One of the most sort of bang you over the head quotes that I've encountered in recent years is, we've moved away from studying human disease in humans. The problem is it hasn't worked. And it's time we stop dancing around the problem. We need to refocus and adopt new methodologies for use in humans to understand disease in humans. This wise person isn't me, isn't PETA. It's the former director of the US National Institutes of Health, a medical doctor who has spent his entire career working on curing human disease. So this is a high-level recognition that we have a serious problem. What HSI has been doing, we've launched a collaboration called Biomedical Research in the 21st Century, or Biomed 21, and we've been taking this high-level concept and working with scientists who have been, again, career in the trenches, Alzheimer's disease, autism. They're in the labs. This is their life. This is why they get out of bed in the morning. And we've commissioned them to do critical reviews objective, independent of us, and what is working, what is not working. And I don't have time today to get into the nitty-gritty of what they found, but these people are shredding the preconceived notions within the science community, and they're doing it on a disease-by-disease basis. And we're taking those results and giving them to the charities who are working in these areas and say, excuse me, can we have a conversation? Because this isn't benefiting your interest, your stakeholders either. We've also been working, as our name suggests, internationally, and we've been having workshops with science funding agencies across the globe. We've had Europe, Latin America, North America, and we're looking at Asia Pacific this year to really have focused debates around what can we do differently? How do we not keep doing the same thing again and again? And it's hard to really measure how successful or not successful some of this is, but this is a PowerPoint slide from the European Commission, Directorate General for Research. And this is the big pot of money for the European Union, 20, soon to be 27 countries. And the way they've broken this down is, I think, quite telling. They're committing to eliminate poorly predictive models in exactly the same diseases that we have handed to them, developing improved models, and specifically replacing animals with better in vitro cell-based or in silico computational tools in exactly these areas. So this is the second largest pot of research money in the world for biomedicine, and they're showing signs of beginning to make that course correction. So again, going back to an earlier question about what are the alternatives. Firstly, I should say I almost never use the word alternative anymore, because what we really, I found using the language of science to talk to scientists is much more effective than leading with the animal card. Because again, these people get out of bed in the morning. I'm a cancer researcher. Avoiding animal use, that's laudable, but that's not my jam. So speaking to them and just taking the same ideas, but reframing, repackaging, rebranding them using a little more scientific lingo has worked wonders in opening doors. So I'm going to be a little geeky here and show you some science pictures, which I find exciting. I hope you do too. But looking at in vitro, again, people think test tube or petri dish, and that's true. That's sort of the old school. We've now, if you look at that little panel on the right, that is what's called a well plate, and that has 1,536 little grooves in it. So think that many test tubes, which no human being, imagine doing that for your daily life. So these robots on the left automatically drop chemicals in. So you can look at a greater number of chemicals, a greater number of dosages, completely automated, work nights and weekends. You don't need to go in and clean cages. So the cost involved in this crashes. The time involved is a fraction of doing an animal test. And the amount of information you get is, you need supercomputers to deal with it because it's information overload. Slightly less efficient, but also much more relevant. We have, if you've heard of organs on a chip, this diagram is from the NIH, the National Institutes of Health in the US, which has invested over $200 million in the past 5 years in this cutting edge, creating a human organ on a chip for key body systems in the human body. This, the top right, is a lung on a chip. So think like a little thumb drive where you have channels for pumping air in and out, so it contracts and expands the way our lungs do when we breathe. And there's a channel for air, there's a channel for blood, and just think if you can study emphysema to lung cancer using human cells that not only sit in a petri dish and get bored and cranky because there's nothing to do. Here they have a much more biosimilar kind of environment where you have, oxygen in, waste goes out. It's much more representative of the real world. Here's a little video. This is a blinking eye on a chip from the University of Pittsburgh. And again, that's human cornea eye tissue. The image on the left is a Drey's test, the rabbit test from the 1940s. It's no contest. I mean, this isn't just an animal person saying, oh, we have better alternatives, trust me. You know, this is the largest funding agencies in the world are putting serious bucks into doing this. And this, right now, this is one organ at a time, but the objective is to have an entire human on one of these tiny little chips that have the key body systems, the blood flow, the oxygen flow. This is where we're close. These are not cheap. These are not fast yet. This is new technology, but this is where the science is going, and the ability to completely strip away the extrapolation issue is, I can almost taste it. And finally, when I think about it, when I hear the words animal model, I think black box. You force feed something to a rat, you know, feet up, feet down, you don't really know what happened. But we have decades of medical research information that is published in science journals, which are behind journal firewalls, where you have to pay and you have to, you know, clunkily go through them and read paper by paper. It's really hard to access what do we, you know, as the human species actually know. How do we capture scientific knowledge? And this is a simplified way of saying from the time we're exposed to something, what do we know about what happens within the molecules in our cells, the tissues, the organ, all the way up. And there's a lot of different places where we can get that information, from clinicians to molecular biologists, and different models that I've covered that can be used to test. This is a really oversimplified way of presenting, you know, this kind of diagram. Human biology is not simple. If it were, we wouldn't be having this conversation. We'd have cracked it. It's not as simple as building a car to replace a horse. This is a grand challenge of our time, and this is why we need very sophisticated computers as well. But the opportunity to move away from this one-to-one animal versus alternative kind of thinking, which has never really worked, and be able to put all of these tools together, we're already seeing some of this being used for safety decisions today. Drug companies are at the very bleeding cutting edge of this. They're using it, they're making decisions. It's a matter of mainstreaming it, making sure that laboratories across the world have this kind of capability. And right now, if we look at the top right, the current approach is expensive. It uses 150 million animals per year. It takes a long time, and the relevance of the predictions to you or me is generally quite low. Again, that 95% number from the drug sector. We're not going to get to zero animal use overnight, but we're on the road. And as we move through it, the cost will go down, the human relevance will go up, and our hope and our belief is that animal use will be reduced to zero ultimately. So to summarize, there are sort of two takeaways that HSI has taken on board in terms of how do we tackle this, how do we not do what isn't working and really, really course correct. And the first, in relation to the testing, is really investing in these mandatory alternatives laws so that as we have these new methods coming on board, hopefully it'll be an automated process where governments just take it and run, rather than we have to, you know, fly to Seoul, fly to Beijing, get them in a headlock and make them, not that we do that, but figurative headlocks. And then the other piece, this is around 10%, I should say, of the body count, is toxicology testing. 85 to 90% is research. The beauty with research is if we can move the money in really big pockets, we don't need to change regulations. We don't, you know, scientists will follow the money wherever it happens to lead. We just have to get better at moving that money. And this takes me back to Liz's presentation around how do we become more relevant? How do we make our voices matter more? And by being politically active is a really key piece. So my takeaway is 3 parts. The first, the easiest, help us ban animal testing for cosmetics in Canada. We have an online petition at becrueltyfree.ca. If you haven't signed it, I encourage you to register to obtain updates from us, because we are entering a hot and heavy period of activity, and your members of parliament need to hear from you. The second, take a stand. The animal research is funded almost exclusively by our money, whether that's our donations or by our tax dollars. And that's, I know, a hard thing to kind of drill into. Charities are easy. Before you make a donation, pick up the phone. You can ask some really pointed questions. You can also reach out to your member of parliament who, that's not going to change the paradigm. It's not going to change the world. But I guarantee they never have anyone call them, email them, write them a letter to ask about this issue. And every time someone does, it's a holy shit moment for them because they don't know what to do. They don't have talking points. They're not prepared. And if everyone in this room reached out to their MP today or tomorrow with those questions, all of this is going to go back to the funding agencies to say, what do we say? We're suddenly getting these questions. What do we do? And just that sensitized activity, and if we can continue to grow that, it is going to make a difference because we have this kind of a schism in our own minds where, it was said earlier, we want treatments for these diseases, we want safe products, we don't want animals to be used, and we need to be challenging that in ourselves, but also in our policymakers and our political officials. And lastly, being a more informed consumer, we're all aware of cruelty-free cosmetics, but how many people are aware that anything that has a germ-killing label claim, whether that's a hand sanitizer or a countertop sanitizer, is legally a pesticide, which means it's not optional animal testing, it is legally required. And that's tip of the iceberg. So the things that we encounter, the things that we buy every day when we go shopping, do we really understand the fine print about the animal testing consequences? Probably not. I don't know why anyone would go looking for that. That's not nice information to have in your brain. But if you go to hsi.org and animal testing, we have fact sheets that will give you some of that fine print. So with that, I will close. Again, all of this change is really incumbent on each of us as individuals and as supporters of Animal Alliance and other animal advocacy organizations. The change that we achieve is all thanks to you. So thank you all for being here.